Students, now we'll start with this new chapter, locomotion and movement. And under this chapter, first we will see the muscles. So here we can understand that it is composed of mesodermal cells and it contributes around 40 to 50% of the total body weight, and it is having several kinds of properties such as excitability, contractibility, extensibility, and elasticity. So altogether it helps in the movement of the muscles, and on the basis of location, these muscles are classified into three categories. First one is called skeletal muscles, second is called visceral muscles, and third is called cardiac muscles. Out of these three, the skeletal muscles are voluntary muscles, and visceral and cardiac muscles are involuntary muscles. So first we'll see what is the characteristics of these skeletal muscles. So these are also called as striated or stripped muscles, and they help in our locomotion. Means our body's locomotion is regulated by skeletal muscles, and by the action of these skeletal muscles, our skeletal system moves and locomotion takes place. And in case of visceral muscles, these are involuntary muscles, and these are also called as smooth muscles, and they are located in the inner walls of the visceral organs of the body, such as alimentary canal and reproductive tract. So if it is present in the alimentary canal, then it helps in the movement of the food, and if it is present in the reproductive tract, then it helps in the movement of gametes. Now the third category is the cardiac muscle. These are again involuntary muscles, but they come under the striated muscles. These muscles are forming the muscles of the heart. That's why it is called, these are called as muscles of the heart, and these are branch muscles. Now we will understand the skeletal muscle structure in detail. Now we'll understand the whole skeletal muscle in detail here. So we can see that several of the bundles or muscle fibers are there, and these bundles of muscle fiber is enclosed in a structure which is called as fascicles. So these fascicles are nothing but the muscular bundle, and these fascicles are formed by the rod-shaped structure which is called as muscle fiber or muscle cells. And these muscle fibers are made up of a plasma membrane which is called as sarcolemma, and under this sarcolemma there is a cytoplasm which is called as sarcoplasm. So the muscle is made up of muscle bundles or fascicles, and these fascicles are present in number, and these fascicles are arranged in a collagenous tissue which is called as fascicle, and each muscle bundle contains number of muscle fibers, and each muscle fiber is covered by a plasma membrane which is called as sarcolemma, and under this sarcolemma there is a cytoplasm which is called as sarcoplasm, and the muscle fibers have several nuclei. So this is the syncytium condition, and endoplasmic reticulum of the muscle fiber is called sarcoplasmic reticulum, which contains several of the calcium ions, and these calcium ions are helping in the movement of skeletal muscles. So we can see here that the parallel arrangement of muscle fibers are forming the myofilament or myofibrils, and several of the fibers are covered in a fascicle, as I told, has alternate dark and light band. Means the muscle fiber is having dark and light band, and these light and dark bands are forming the striated appearance. On the basis of that, we used to say these striated muscles, and this pattern of light and dark band is done, or we can say is created by actin and myosin proteins. So these are main two proteins which forms the light and dark band in the muscle fiber. So we can see the details of this actin and myosin here. So these actins are the proteins which are thin and the myosin are thicker. So since these actins are thin, so it gives the light band on muscle, and this light band is called I band or isotropic band. And the myosin, as I told, is thick in appearance, so it gives the dark band in the muscle, and this band is called A band, and this A band is formed by the word anisotropic band. So now we can see the structure here. So by this figure, you can understand the structure of one muscle fiber. So you can see this one is the dark band, this one is the light band. So there is a pattern of dark and light band, and this light and dark band is regulated, or we can say represented, by the different kinds of protein, actin and myosin. So we can see here there are several of these muscle fibers, and these muscle fibers or myofibrils are composed of actin and myosin rods. By this figure, we can understand how the darker and light bands are forming in skeletal muscle. So we can see this rod-shaped structure, which is thick in appearance, is representing the myosin protein. So as I told, that myosin gives the darker color. So starting from this region up to this, means from here to here, it will show the darker band, just because here the myosin is present, and it doesn't depend the presence or absence of the lighter band. If there is a darker band, or if there is the myosin, then it will represent the darker zone, and this zone is called A band. So we can see here this zone is representing A band, and due to presence of myosin, it represents the darker band. However, starting from here to here, there is only actin protein. So as I told, that the actin is a protein which is thin as compared to myosin, so it represents the lighter band. So wherever the only actin is present, then it will represent the lighter band, and this band is called I band. So we can see that from here to here, the myosin is present. It means it will represent the dark. This region from here to here, there is only actin, so it will represent the lighter band. After that, again myosin comes, so it will represent the darker band. After this myosin, again there is the only myosin, so again it will represent the lighter band. After that, darker band. So it gives a lighter and darker zones in the muscles alternatively, and it gives a characteristic feature in the skeletal muscle. That's why it is called as striated muscle. Now you can see that there is H zone also. It means H zone, you can see H zone is present here, means where only myosin is present. H zone is the zone where only myosin is present. You can see that here there is no actin. A band, as I told, wherever myosin is present, either actin is present or not, then all the region where myosin is present will be called as A band, and all the regions where only actin is present is called I band. And here there is a Z line, and this Z line is representing the end of one muscle fiber. So you can see these are two Z lines, first Z line and second Z line, and in the middle of this structure it is called as M line. So A band is representing myosin, and there is means actin may be present or not. I band is representing only actin, means there is no presence of myosin. Z line present between I band. So you can see the I band, this is the I band, and in between the I band there is a zigzag structure. That's why it is called as Z line. And from this zone Z line to Z line is representing one myofibrin. So you can see this structure. From this zone to this zone is called myofibrin. Now we'll see the details of actin and myosin protein in detail. So structure of contractile protein is the main heading, and under this heading we will understand the actual, or we can say the zoomed structure of the actin and myosin protein. So as I told before that actin is a thin filament. So this actin is composed of two kinds of components. First component is called tropomyosin and the second component is called F-actin. And here both of these components are present in pairs. And here both of the components are rope-like structure and they are helically wound to each other. And in case of tropomyosin, as I told that it is present in pair and it is helically wound structure and it is made up of monomers. These monomers are called as G-actin. G-actin means globular actin protein. And the F-actin, as I told, that it is again present in the pair and these are also helically arranged structure and these are filamentous structure. This F-actin is the filamentous structure. Means all these two pairs of tropomyosin and F-actin, they are again helically arranged to each other. Means a total of 4 rope-like structures are there which is forming the actin protein. And there is one more protein which is called as troponin. And this troponin is a complex protein which is distributed over this actin, or we can say the 4 helically arranged structures at a regular interval. And it marks the actin binding site of the myosin on the actin filament. So we can see the whole structure here. So we can see this structure. These rope-like structures, these are helically wound rope-like structures are called as tropomyosin, and these bead-like structures are again forming a helical structure and it is again present in the pair which is called as F-actin. Now we can see at the regular intervals troponin is there. So troponin is a complex protein, as I told before that it is present at the regular intervals on these actin filaments. And what the important point associated with this troponin is it marks the activity of binding site of myosin on the actin filament. It means it marks the active site, means where the myosin gets adhered or attached to this actin will be marked with this protein complex. Means during the movement of the protein or we can say the filament of actin over the myosin will be regulated by this complex proteinaceous structure which is called as troponin. Now we will see the detailed structure of the myosin. And as I told before that myosin is a thick filamentous protein and we can see the structure. So here we can see again there is a 2 helically arranged structure which appears like a rope, and at the end of both the rope there is a lobular or button shaped structure. So we can see these rope-like helically arranged structure, and these helically arranged structure which is present in the pair is called meromyosin. And this is we are just seeing the tail of this structure here, and this tail-like structure, or we can say the end of this myosin is represented by the lobe-like or button-like or knob-like structure, and these are called as cross arms. So we can see these cross arms, and these cross arms are having head-like structure. We can see here that head and cross arms means this the end or tail of these myosin is composed of two important structures which is divided into head and cross arms. And over this head there are two different sites. First site is called actin binding site and the second site is called ATPase enzyme site. So we can see here this head is having two important sites, actin binding site and this ATPase enzyme. Now the same thing has been written here also, that there are the tail, there are the cross arms and there are heads, and these heads are globular structure, and these head and cross arms are heavy neuromyosin. Means this structure, the rest of the rope structure from here up to the another end will be called as light neuromyosin. Means it has been formed by light neuromyosin chain. However, this structure means cross arm and head, it is formed by the heavy neuromyosin, and it projects outward at regular distance from the myosin. Means in the myosin there are several of these pair of ropes which is most made up of light neuromyosin and heavy neuromyosin. Light neuromyosin is forming the whole structure, means the whole straight structure, but at the end it forms a head and cross arm, and this head and cross arm is formed by heavy neuromyosin structure. Now we'll see the mechanism of muscle contraction. But before understanding the mechanism of muscle contraction, we need to understand this structure again, this myosin structure. So by this figure we can understand easily how the myosin look alike. So we can see these are the paired structure which is forming the myosin, and we can see that several of this kind of myosin paired structure is forming the thick filament, and as I told that at the end of these filaments it protrudes out. So we can see several of this kind of head and arm, cross arm is protruding out from the main structure. So we can see several of this kind of structure is protruding out, and this protruded structure bearing the head is having the ATP binding site and ATPase enzyme site. And this figure is representing the actin filament, and as I told before that there are two kinds of pairs of the structure. So one kind of pair is here and another kind of pair is here, and this one is the blue colored structure which is present at the regular interval on the thin filament is the troponin. So we can easily understand this structure. And this figure is representing a single myofibril. So we can see here several kinds of dark and light bands are present here, and we can see here one myofibril segment here. This one is the one Z line and this one is the another Z line, and in between these Z lines this is the zoomed structure. We can see that this blue colored structure is representing the actin filament, you can see here, and this red colored structure is representing the myosin. So we can see that myosin is there and actin is there, and myosin is the structure. Over this structure we can see actin is present, and actin slides over the myosin. So by the sliding of these actin over the myosin helps the contraction and relaxation of these proteins, or we can say ultimately the muscle fiber. So what happens when this Z line will go towards this M line, then what will happen? If there will be the contraction, but what happens when this Z line is going on opposite side of this M or middle line, then it helps in the relaxation of the muscle. Now we will understand the mechanism of muscle contraction, and this mechanism of muscle contraction is taking place in four different steps. So by this figure, we can understand that this one is the portion of the actin filament and this one is the portion of myosin filament. And as I told before that actin filaments move or slide over the myosin. So we can see here these are the wrapped structure and this one is the troponin and this one is the small portion of the myosin. So one by one we will see the step by step processes. So in the first category or we can say the step of this muscle contraction is the binding, and during binding what happens, so the neurotransmitter in the form of acetylcholine is released on the neuromuscular junction or neural plate. So what happens when the signal is sent from our brain to the muscle, then the finally, as I told before, that the chemical message is transmitted from the the end of the neuron to the muscle is in the form of a chemical, and this chemical is a neurotransmitter is acetylcholine. So what happens? The acetylcholine is released over the neuromuscular junction and it generates the action potential to the sarcolemma. As I told before, what is sarcolemma? Sarcolemma is the plasma membrane of the muscle fiber. So what happens when this neurotransmitter is released on the sarcolemma? Then first step of the mechanism is the release of calcium in the sarcoplasm. So what happens when the calcium is released in the sarcoplasm? Then the calcium binds to troponin on actin filament, thereby remove the masking of active site of myosin. So as I told that these troponins are present on the actin and it masks the active site of the actin. Then what happens when it reacts when the troponin reacts with the calcium, then the active site is exposed by the removal of this troponin. Then what happens? Simultaneously, the hydrolysis of ATP takes place. So as I told, where the ATP is present, ATP is present over the head of myosin. So hydrolysis of ATP takes place. So what happens? The energy will be released. Then the second step of the this mechanism takes place, which is called as power stroke. So under this power stroke, the myosin head now binds to the exposed site of actin to form the cross bridge. So what happens when the troponin is removed? Then the active site over the actin is exposed. Then this head of this myosin gets attached with this active site over the actin. So it forms the cross bridge, and the formation of the cross bridge is taking place in the step which is called as power stroke. Now what happens next? Then the movement of actin filament takes place towards the H zone. So what happens? This one is the actin. So at the regular intervals, troponin is present. When the troponin is removed by the reaction of calcium, then the active site will be exposed. Then what will happen? The head of the myosin will get adhered to this actin, and it will help in the pulling of the Z zone towards the center. So what happens? The movement of action actin filament towards the H zone takes place. Then shortening of the muscle fiber takes place. Or what happens when the shortening of the muscle fiber takes place? Then I band, which is representing just the myosin, will be reduced. And what happens next? The next step is called detaching. So what happens when the actin will slide towards the M zone or the middle zone? Then the head will be detached. So now the ATP binds and the cross link structure will be broken. So the breaking of this head with the myosin will be called as detaching. Then what happens next? the resting phase occurs. So when the calcium is pumped back, when the movement will be completed, then when the calcium will be pumped back to the sarcoplasmic cisternae, resulting in the masking of the action filament. Then what happens again? When the calcium will be pumped up back, then there will be less calcium. Then what will happen? The troponin will again mask the active site of the actin. Then what happens? It reverts the Z line back to the original position. Now the relaxation takes place. So all these four steps is explaining how the sliding of actin takes place over the myosin band. And to understand completely, we can see this video and we can understand the whole mechanism by this animated video. So we can see here that, as I told before, this red color structure is representing the actin. This structure is representing the myosin. This structure is representing the Z band. These structure where the only actin is present is I band. A band is there where the only myosin is present. Now we can see the H zone. Now the actin myosin binding cross-linking takes place. We can see here several of the heads are getting attached with this structure. Now the sliding of filament takes place. Now we can see how this attachment and detachment takes place and how the actin moves towards the center. Now we can see the yellow structure is representing the myosin and the red structure is representing the actin. Now we can see how the movement is taking place. Now by the another video we will understand the same mechanism in another way. Now we can see how this movement takes place. Now you can see this actin is moving over the myosin. You can see how this contraction and relaxation is taking place. This thick structure is representing the myosin and this structure is representing the actin. Now you can see several of the myofibrils, how they are contracting and relaxing. And by the contraction and relaxation, or we can see the sliding of the actin, the contraction and relaxation of the muscle takes place. Now it is representing the single unit.